Very much. I realize I'm standing between you and lunch, so I'll get cracking. So thanks very much to the organizers. It's been a great meeting, and thanks for the opportunity to discuss this work. Thanks also to the Biochemical Society for contributing to my trip here. So I'm interested in understanding the molecular mechanisms that link aging or resilience. We had a nice talk from Helen Graves earlier. And the mechanisms linking aging resilience with dementia. And the spoiler is that it's all about the microglia, obviously, which is why we're all here. And more importantly, how microglia interact with other cell types, particularly neurons and oligodendrocytes, as you'll hear about in the talk. So I think it's important to do this in an unbiased way, and the way I do that is to work mainly at the transcriptional level. And so we've integrated data from a variety of models, from mouse models of Alzheimer's and aging, human pathology tissue, and a variety of cultures, and more recently, iPSC, human iPSC differentiated to microglia in collaboration with Jennifer Pocock. And the overall goal of this work is to identify new targets for drug discovery and to be able to predict disease risk better, with the ultimate goal of boosting the beneficial effects of microglia. So we're all familiar with the Alzheimer's cascade, so I won't dwell on this, but familial and environmental factors lead to elevations in A-beta, and then over the course of a couple of decades, A-beta converts to tau pathology, and it's the tau tangles that cause the death of neurons. So my first entry point into this work was working with John Hardy to discover basically where the late onset Alzheimer's risk genes that were expressed by microglia, where they fit into this Alzheimer's cascade. So the initial amyloid cascades, it was all about the neurons. An obvious omission was the importance of the innate immune system. And with the discovery of these genes, the project that we did with John was basically to understand where these genes might be functioning in this cascade, and I'll show some of the data in the following slide. But we saw that these genes were likely regulating the transition via microglia between A-beta and tau pathology. Bart Destruper has got similar data based on that. We've heard a really nice study from Yaming Wang as well, which elegantly shows that the risk genes are involved in conversion of A-beta to tau, and there's also similar mouse data from David Holtzman's group, and also from David Hansen at Genentech as well. So the model that we have now, if we integrate the GWAS, so with GWAS, we probably predict that there's around 150, 200 risk genes that contribute to risk of dementia. So if you have a good collection of SNPs, you're going to move down the amyloid cascade slowly, and you'll live a long and happy life and probably avoid dementia. If you have a bad collection of SNPs, you're going to move down the cascade more quickly, and in your 70s and 80s, you'll show signs of dementia. Age also obviously plays a role in this. We don't understand the mechanisms. The older you are, the more likely you are to reach the end stage of neurodegeneration. And so coming back to approaching this question in an unbiased way, GWAS have been remarkable in that they've completely changed the way that we think about Alzheimer's, but there are still important limitations to GWAS as well, and those are discussed in this nice paper by my collaborator, Valentina Escott-Price. So there are several limitations to GWAS. What's relevant for today's discussion is that if a SNP or a variant is involved in multiple diseases, then that person is less likely to make it into an Alzheimer's GWAS. They'll get other diseases before they reach that age. And so those SNPs and variants are going to drop down this p-value of significance. And so what we wanted to do with John and Valentina was to mine these subthreshold variants between these two values of significance to see if we can gain new insights into disease. And so the way that we did that is to mine the subthreshold variants from GWAS. So we've looked at Alzheimer's GWAS, and we've published that work. And what I'm talking about today is the longevity GWAS. And we're filtering those subthreshold variants with transcriptional changes from bulk and single-cell RNA-seq from human and mouse models. So in the lab, we're also working on increasing our... sorry, the slides are being slow here. So increasing our omics capabilities and then integrating epigenetic data and spatial transcriptomics as well. So, and again, ultimately, this is to understand disease risk better for prediction, provide new drugs for drug discovery experiments. So As a proof of principle, this was the study that we published where we're basically mining Alzheimer's GWAS with transcriptional changes in the old generation of amyloid transgenic animals and tau transgenic animals. I'm not going to talk so much about the tau, but the data is published, so you can look at that later or we can discuss that at lunch. So with RNA-seq, you get a lot of data, and making sense of that data is always a challenge. And so we use a bioinformatics approach called network analysis to classify gene expression by their gene expression patterns. And so we can quantify the pathology, the amyloid pathology, nicely in the mice with age. And we can see that TREM2 has got a very similar expression pattern to the amyloid pathology, as does this antiviral enzyme interferon responsive OAS1. And so the network analysis gives us around 1,500 genes, that have this expression profile. Those are represented here. It's not a surprise that these are expressed mainly by microglia, by the innate immune system. And so we call this the amyloid responsive microglial network. Within this network, we saw that there was an enrichment of known variation associated with Alzheimer's. So everything that's marked with a red oval here is a known risk gene for Alzheimer's. So TREM2 is there, it's a hub gene. The hub genes are thought to drive the cellular response to amyloid, CD33, SPI1, so some of your favorite genes are there. And so what we do with this analysis is when you do a GWAS, you have to correct for 40,000 genes for 40,000 comparisons. Here we've narrowed down the genes of interest to 1,500. We're correcting for 1,500 genes. And so these subthreshold variants marked with the blue rectangles now become significant. And so these are putative risk genes for Alzheimer's. OAS1A was one that we've followed up on. It's an interferon responsive antiviral gene. Nadia Magusala has a poster on there, so talk to her, I'll catch her at lunch. So we went on to go from a putative risk gene to showing that it was really a risk gene for Alzheimer's and understanding its mechanism of action in iPSC microglia as well. And we're doing some follow-up work with Lilly now as well to understand mechanisms better, moving towards drug discovery. LAPTM5 is a lysosomal protein, also a putative risk gene for Alzheimer's. So the amyloid responsive microglial network was enriched for genetic variation associated with Alzheimer's, but the tau responsive microglial network was not enriched for genetic risk for Alzheimer's, and that's why we can place the known Alzheimer's risk genes into the transition from amyloid to tau pathology. So that's all published, and our gene expression data is available on this web app as well if you want to look at your genes of interest later. So this is being slow. Okay, it's frozen now. That's not good. So the next slide is the hypothesis side. Hopefully it will catch up as if I keep speaking. So the hypothesis is that genetic variability in the response to cellular damage influences disease risk and longevity. So we've already talked about A-beta pathology, and I'm going to go on to talk about aging. And the aims of the study were, can we understand what genetic variation associated with lifespan is telling us about brain aging? I'm having problems down here. More than usual. Just give you a second and then. Yeah, I can find where we are. Thanks. Okay, brilliant. Thanks. Yeah, so the hypothesis. So the key point is that we're looking at the response to cellular damage rather than what's initiating it. And the aims of this particular study were what is genetic variation associated with longevity, telling us about changes in brain cells? Are there links between variants associated with longevity or resilience and Alzheimer's risk, and does this give us new mechanistic insights? So the longevity GWAS that we're using are from these two published studies from Timmers and Deline, where they take 3 measures of longevity. So the first is surviving to the 90th percentile of life versus the 60th percentile of life, and there's tens of thousands of samples in these. So 90th percentile of life for an average male is 89 years of age, and 60th percentile is 75 years of age. So we're looking at a 14-year gap in lifespan and what genetic variation contributes to that. Second measure, parental lifespan, it speaks for itself. Third measure is health span, so surviving to an old age without obvious disease. And so we used exactly the same approach, the network analysis, to identify genes that are behaving similarly. This time we're using bog standard black 6 mice aging from 2 to 18 months of age, and here are examples of genes that show strong age-dependent changes. The lysosomal protein, LAPTM5, is one of those, as is interleukin-33. So it won't be a surprise to you that the module or the network most associated with aging from bulk hippocampal samples was the microglial network, and this is how it looks. So you'll recognize lots of things here. The lysosomal protein is here, LAPTM5. ApoE is here as well. So ApoE changes, this is telling us that ApoE changes more in microglia than other cell types with aging. What was more of a surprise was that in the center of this network, the hub genes, a number of these were expressed by oligodendrocytes, such as IL-33. And to us, that's suggesting that there's an interaction between microglia and oligodendrocytes, which I'll go on to talk about more. So that was the mouse network. It's shown again here. We've done the similar analysis in human aging samples from the GTEx data set. So some of you will be familiar with this. This is data from around 100 individuals, hippocampal bulk RNA-seq again from a variety of ages. And the network analysis again shows that the module showing the greatest changes with age with a microglial network. This is how the network looks. Again, it's very similar to what we've seen already. TREM2, CD33 is here, TYRO-BP is a hub, LAPTM5 is there, CD11B is there, so no big surprises. The slight sting in the tail is that this network is from healthy aging individuals, so they don't have obvious dementia or obvious cognitive problems. There may be early amyloid deposition, and this in fact looks like the DAM network that we've heard quite a lot about, but this is just age-dependent changes. So we found that the oligodendrocyte network was also strongly age-dependent in humans and in mice. This is how the oligodendrocyte network looks. So I'm showing this figure because it illustrates why I think network analyses are so important. It kind of allows us to look at genes with new insights. Particularly here, we see that presenilin-1 is a hub gene, and so it's driving the oligodendrocyte response to aging, and that was a shock to me because I think most of us don't think about presenilin-1 and oligo function. And so now we have these age-dependent networks. We asked if there was enrichment, and actually we derived several modules and networks that we looked to see if there was enrichment for Alzheimer's risk. And it's probably not a surprise to you that the microglial network was enriched for genetic variation associated with Alzheimer's, even though these samples are from people without Alzheimer's disease. And we also saw enrichment of the oligodendrocyte network with variants associated with Alzheimer's as well. So it's significant, but modest, and the genes marked with the blue ovals here drive some of that risk. So obviously, something else that we've touched on here is similarities and differences between humans and mice as well. So we've done a preservation analysis of these networks, and both the microglial and the oligodendrocyte network, they fall between these dashed lines of significance. So there's modest preservation of these networks between human and mice, and modest preservation means that there's some significant overlaps between the network, but there's also important differences as well, which we shouldn't ignore when designing experiments. I'm not going to talk about those overtly, but we can discuss those at lunch. because there are some interesting insights. So the last couple of data slides now, I'm going to talk about age-dependent networks from single-cell RNA-seq data. So this is from our collaborator, Carla Sala Frigerio. This is isolation of microglia, again, from black 6 mice. So these are just wild-type mice, a variety of ages again. A number of the modules from the isolated microglia were age-dependent. We're going to look at a couple of those. So now we're looking at the amyloid-responsive microglial network, or the DAM network. So we've seen these trajectory plots before in this meeting, where each dot summarizes the expression of a single cell, and in 2 dimensions here, you can basically place the activation status of a cell, and it falls into 3 main categories: homeostatic, amyloid-responsive microglia, and interferon-responsive microglia. So here, with these genes, we're looking at the yellow and orange cells on the amyloid-responsive arm. This is how the network looks. You'll find familiar genes that you expect to see here. CLEC7A, ApoE is here, antigen presentation, CSF1 is here. These network of genes increase with old microglia compared to young microglia. This is a completely independent data set for validation. This is bulk isolation of microglia and RNA-seq. And we found that this network showed a tendency to be enriched for Alzheimer's risk, so it's not quite significant, but this is mouse data compared to human GWAS, and then the coverage of single-cell RNA-seq is low. So another module of interest was this HM2 homeostatic module. So now the yellow-orange cells are on the left homeostatic arm. This is how the module looks. Again, it looks as you would expect. TMEM119 is a hub. P2RY12 is a hub. There's also a few surprises in here as well. So ABI3, the Alzheimer's risk gene, is here. CSF1R is here. And the lysosomal protein that I've discussed previously, LAPTM5, is here as well. So these are thought to be involved in homeostatic processes, and these genes are downregulated in old microglia compared to young microglia. And the reason why I'm showing you this network is because it's increased for genetic risk, genetic variation associated with longevity or resilience. So these variants seem to be affecting homeostatic functions of microglia. And so the final data slide is that we wanted to explore further the links between microglia and oligodendrocytes. And so we turned to this Nugent data set where they take young control mice and they treat them with this demyelinating cuprizone agent. And then they isolated the microglia, did RNA-seq, and we took that data and we did a preservation analysis with our age-dependent single-cell networks. And basically, you can see here that all of the modules are way above this dotted line of significance. So all these modules are highly preserved between aging and demyelination. And we'll look at 2 quick examples. This is the amyloid-responsive network. It increased with aging, and here it's increasing with demyelination. And the homeostatic network decreased with aging, and here it's decreasing with demyelination as well. So what this is suggesting is that with age, microglia are responding to changes in myelination. And obviously now we have a sense of what gene variants associated with Alzheimer's and longevity may be driving these responses. So to summarize what I've told you, we've looked at Alzheimer's variants, and by filtering them with these different RNA-seq data sets, we have around 100 putative risk genes for Alzheimer's disease, and also around 100 putative risk variants associated with lifespan. The Alzheimer's variants seem to affect activated microglia and also oligodendrocytes, whereas the variants associated with lifespan are expressed by homeostatic cells. And these are the genes driving that enrichment. So some genes we know about, and then some putative risk genes that we're following up in the lab to see what they may be doing in Alzheimer's, in microglia and oligodendrocytes. And the variants associated with longevity that are enriched in homeostatic microglia are these types of genes: caspase 8, the FOXO3 transcription factor, which has been long associated with longevity, and particularly in worms and flies. In terms of what's common between longevity and Alzheimer's variants, the overlap was very low, and it was limited to these 3 genes, of which ApoE is not a surprise. And so both of these GWAS are impinging upon microglia at different stages of their life cycle. And we believe that by identifying these variants associated with lifespan expressed by microglia, that we found some of the genetic drivers of inflammaging. And so the hypothesis is that if you have a good collection of SNPs to these longevity variants, you're going to keep your microglia in a homeostatic state. If you have a bad collection of SNPs, they're more likely to escape that homeostatic state. And then a different collection of SNPs in the activated state would then push some people into dementia. So that's the model, and basically in the lab now we're trying to identify what some of these genes are doing to iPSC microglia. So to quickly summarize, I've told you that risk for Alzheimer's is enriched in microglia and oligos. Genetic variation associated with longevity is enriched in homeostatic microglia, and an important age-dependent role of microglia may be to react to changes in myelin status. So this data is available on the medRxiv now, so yeah, it'd be great to get any comments that you have. So this work was led by a brilliant student, Andy Graham, with help from Effie and Umran Yaman, who spoke earlier today, and thanks to my mentors, John Hardy, Valentina Escott-Price, Jennifer Pocock, and thanks very much to our funders and the Biochemical Society again.
Thank you, Dervis. I know we've run over a little bit, so our apologies for that. But are there any burning questions, or do you want to leave it for lunch chat? We've got one in the middle. So.
Hi, this is really interesting work. I was just wondering if you looked at potential gender-sex differences between this network, if there's potentially divergence between genders and what variants are protected, or different sort of networks might be.
Yeah, that's a really good point. We haven't actually done that. That is a really good point. Yeah, we should do that.
Okay. Maybe a quick question. Does your data suggest, Dervis, that targeting oligos then would be a good way to go? It's been done in MS, obviously. But.
Yes, I think so. I think we're more interested in the interaction between microglia and oligos, but yes, effectively. Okay. Yes, I think like oligos have been largely unexplored now in Alzheimer's disease, and I think we need to think about that more in terms of biomarkers for oligos, maybe imaging approaches that look at myelin status more. I think those are the things that we need to work towards.
Okay, great. Well, thanks, Dervis, again, and thank all the speakers for this session. So thank you.